That is pretty impressive because, I mean, I can't get anywhere without Google Maps. So,
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
and I am David Peterson, your host for today's episode. For the past couple of years, moviegoers have thrilled to the sight of sandworms large enough to swallow a spice harvester. But today we'll talk with a researcher who studies far smaller and probably far deadlier worms.
Our guest today is Dr. Astra Bryant, assistant professor in the Department of Neurobiology and Biophysics at the University of Washington. We were set to interview Astra in the podcast studio as part of her visit for the CTEGD Symposium, which we hold annually in the spring at UGA. Unfortunately, we ran into some issues, so here we are. Astra is a neuroscientist and a parasitologist specializing in the thermal physiology of soil-transmitted parasitic nematodes. Astra, welcome to the podcast.
Great to be here and great to have the chance to speak with you about the worms.
So, Astra, one of the first questions that we usually ask our guests is to just give us a walk through what your pathway into science looked like.
I'd say that my pathway began with an early love of animals and me being convinced for many years growing up that I was going to be a large animal veterinarian. That certainty lasted all the way through about midway into my undergraduate education when I had the opportunity to work in the Philadelphia Zoo in the winter. And I discovered that being outdoors in the cold isn't my natural habitat. And my natural habitat is much more inside the lab. At that point, I started working in a neuroscience research lab and fell in love with brains and with doing research and asking questions, which to me is the fundamental part of life as a research academic. And I've been following that path ever since.
Well, I think that leads right to our next question, which is, what is the fundamental question that now drives your research?
So I'd say that writ large, I'm driven by a curiosity about how the brains of parasitic worms have evolved to enable these truly fascinating creatures to perform parasitism. My background is in sensory neuroscience, so I've always been really interested with how animals experience the world around them and how they translate their knowledge of the world as comes in through their senses into essential behaviors. And in the context of parasitism, this leads to a fascinating merge of neuroscience and parasitology, and in trying to understand how these tiny organisms can move. It leads to this fascinating synthesis of neuroscience and parasitology in trying to understand how these very small organisms can find and infect a very large source of neglected human disease.
So you mentioned a key word there, which is sense, and you've already told us that you're a neurobiologist. So these tiny little creatures, which I imagine have an equally tiny brain, are able to sense a host. How does that work?
They can sense a host, and the answer to the question of how they're able to do that, and in fact, the answer to a lot of questions about parasitic worms that we don't know. We don't know a lot of things about these worms. Their basic biology is very poorly understood. What we do know is that the brains of parasitic nematodes, like other nematodes, are approximately 302 neurons, which is about, I'd say, a a fifth of the total number of cells in their body. So their brains are a pretty sizable chunk of real estate. And nematodes, including parasitic nematodes, can sense a lot of things about the world. They seem to be able to sense different chemical cues. They can sense vibration. They can sense carbon dioxide and oxygen, and they can sense temperature. And it's their thermosensory abilities that I study and that I find particularly intriguing.
So the particular worm that you use, Strongyloides stercoralis, tell us a little bit about it.
Yes, Strongyloides stercoralis is a soil-transmitted parasitic worm that is estimated to infect approximately 610 million people around the world. It's endemic to continents, including Asia, Africa, South America. And truly, they're a disease of poverty. So you can find them not only in those continents, but also really in any community that has poor sanitation systems. In recent years, they've been found in poor communities in various states within the United States, including Texas. So these worms have a complex life cycle that is similar to other parasitic worms that maybe folks have heard more about. So things like hookworms, I think, are maybe more in the public consciousness. Strongyloides stercoralis is very similar to hookworms. Like them, it's infects primarily through skin penetration. Once it gets inside the host, these worms migrate to the small intestine where they develop into parasitic adulthood. They reproduce in the small intestine, and their eggs and larvae are voided out of the host on feces, where they develop back into infective larvae that seek out hosts and restart this cycle. In the past few decades, Strongyloides stercoralis has been rising to prominence as an emerging model system for other parasitic nematode species, such as hookworms. And that's because Strongyloides species, so these are species in the genus of Strongyloides, have a unique aspect of their life cycle. In the past few decades, Strongyloides stercoralis has been rising to prominence as an emerging model system for other parasitic nematode species like hookworms. And that's because species in the genus Strongyloides have a unique aspect of their life cycle. The eggs and larvae that emerge from the host, these are the progeny of the parasitic adults, in Strongyloides species can cycle through a free-living generation. This generation includes free-living adults, both males and females, who produce progeny that all become infective larvae. And what's important about these free-living adults is that morphologically, they're very similar to an extremely common model system, Caenorhabditis elegans, or C. elegans. And so the C. elegans field for a very long time has been developing an exquisite set of functional genomics tools. And because Strongyloides species have this free-living life stage, we can apply those tools to the parasites. And this life stage, as I said, is unique for Strongyloides, so we don't see it in hookworms.
So C. elegans I've always found fascinating because, as you mentioned, the detailed genetic work they've done, I think they've actually knocked out individually most of the genes, which is amazing. But if we were to take a cell map of C. elegans and match it with a cell map of Strongyloides, how closely does it match up in number of neurons and the size of the brain and everything else?
It's a great question and one that I would love to know the answer to. Let me back up to give you the context for the scale of knowledge that we have for Strongyloides species compared to C. elegans. So C. elegans, there are thousands of labs studying this worm and studying questions using this worm around the world. For Strongyloides, there's been, by my count, less than 5 labs that have ever maintained them in a laboratory environment. So we're a little bit further behind. We've made many strides in the past few years to start adding tools, functional genomics tools, to the Strongyloides research field. But we're still so far behind that it's hard for me to answer that question. What we do know is that if we just line 2 worms up, C. elegans and Strongyloides, there's a lot of things that look similar about their basic anatomy. One of the things that my lab is actively working on is trying to understand how the conserved nematode nervous system is adapted in parasites versus C. elegans to produce the very different behavioral repertoires of these 2 species. And part of that is trying to identify individual neurons that are conserved between these 2 species. And in general, as I said, from a sort of far out picture, there seems to be many similarities. We sort of expect the same number of neurons. They appear to be in approximately the same configuration, but we know there have to be differences because C. elegans is not a skin-penetrating human parasite.
So even though it's not a skin-penetrating parasite, does C. elegans have any heat-seeking behaviors?
I'm going to reframe that question a little bit. The answer is yes, but a better question to ask is, can C. elegans sense temperature at all?
Okay.
The answer is yes, they absolutely can. C. elegans has an exquisite repertoire of thermosensory behaviors. Research from many, many labs over the decades has shown that C. elegans uses its ability to sense temperature to navigate to a physiologically and reproductively permissible temperature. In general, that's around 15 to 25 degrees Celsius. And they have, as I said, these exquisitely complex behaviors. They're extremely sensitive to thermal gradients. What they don't do is display the kind of attraction to body heat that we see in the infective larvae of mammalian parasitic worms. This is true of both Strongyloides stercoralis, as well as a couple of other species, including a human hookworm. These infective larvae, which are very similar morphologically to the dower larvae of C. elegans, so both of these are developmentally arrested, they're found in the soil. The infective larvae show this very dramatic attraction to host body temperature. They're moving quite rapidly towards heat. And it's pretty likely that this is one of the behaviors that enables these worms to actively locate hosts in the environment, in combination with their other senses.
So the basic sensory parts are there in C. elegans. Is when we get to stercoralis, they're wired a little differently. The programming is different.
That's the strong hypothesis.
Okay.
So far, what we have published is we've identified the sensory neurons in Strongyloides that are responsible for the attraction to body heat. These neurons are called AFD, and they're conserved between C. elegans and Strongyloides. And in both of these species, they're the primary thermosensory neuron. So you have these radically different behaviors and the same primary sensory neuron pair. What we don't know is where does the difference in preference arise? It's very possible that that's due to differences in the wiring further on into the Strongyloides brain. We do also know that there's additional adaptations. We've found some of them, and they include things like the thermosensory proteins that act in these cells, these AFD cells. In Strongyloides, stercoralis are much more tuned for mammalian body heat, as opposed to their homologs in C. elegans. And in fact, exploring that is one of the things that my lab is currently working on, trying to understand the evolution of thermoreceptors in nematodes to see, is this shift towards sensitivity for mammalian body temperatures something that you see in other species like hookworms? And to be clear about that question, one of the things that is most fascinating to me about parasitic nematodes is that in Nematode, it's estimated that parasitism arose at least 15 times independently across evolutionary history. So when you look at Strongyloides stercoralis, that's a different evolution of human parasitism compared to human hookworms. So you have these two species that independently decided, hey, penetrating the skin of a human and migrating to the small intestine is a reasonable life choice. And for me, the question is, has evolution converged on the same adaptations to allow these two species to accomplish this really remarkable feat for a nematode?
That's really fascinating because hookworm and Strongyloides get just grouped together under soil-transmitted helminths. And so I assumed that they were related, but I guess on a phylogenetic tree, they could be fairly different and separated by non-parasitic ancestors, different ones.
That's correct. It's hard to estimate whether parasitism evolved as the ancestral trait or not. The fossil record for nematodes is let's say, non-existent. But it's definitely true that our understanding is that strongyloids evolved independently from hookworms. And so I'm really fascinated by this concept that you have all of these converging species that all show very similar behaviors, but may or may not be accomplishing those behaviors through separate molecular and cellular mechanisms.
So you just talked about behaviors, or you mentioned behaviors. I'm not sure what any of our listeners or even myself even thinks of when they think of worm behaviors. Can you give us an example of what we're talking about here and what it means to quantify them?
Yes. So there's two, let's call it behaviors, that I'd like to talk about. The first one is the migration of these infective larvae towards, in the case of my lab, heat. So what I want folks to imagine is that you have a bunch of tiny worms. These worms are 0.5 millimeter long and about 1/10 of a millimeter wide. So tiny. And what they're doing is they're on a surface, let's say it's soil. In the lab, we have them on an agar sheet. And in response to a thermal cue, in the lab, we have a machine that enables us to produce a thermal gradient. What you'd see is that these worms all orient and sort of swarm towards the warmest temperature they can possibly sense. These worms they're crawling and they're moving relatively quickly, particularly for a worm. I'd say that about 20 centimeters worth of movement is probably going to take these worms on the order of 5 to 6 minutes, which sounds sort of slow, But remembering that these worms are 0.5 millimeter long, they are absolutely booking it. I've, for a long time, described this heat-seeking behavior as the most robust sensory-driven behavior we've ever seen in any nematode species.
So one thing that I find really interesting in acquiring their target, moving toward it, they need the thermosensing. As soon as they get inside the host, they have to turn that off and turn on some other sensory behaviors so that they get to the gut. Do we know anything about this transition?
Yeah, very little. What we do know, I should make it clear that thermosensation is definitively not the only sensory cue that these worms are using as they are locating hosts. There's a host of others, pun not intended. The question of what they do when they get inside the host is a really, really good one. In general, we can break it down into two questions. One question is a very general one of how does this worm survive the transition from the soil environment to the host environment? And maybe more specifically, how is it possible that these nematodes are able to survive inside our body heat, which is a temperature that C. elegans can last a couple of hours in? And yet, the parasitic adults of Strongyloides stercoralis can live for many months inside the host body. So we don't know what's driving that ability, which appears to be acquired. So there's a manuscript that my lab has just submitted for publication where we looked at the free-living life stage of Strongyloides stercoralis. You will hopefully remember these from earlier in our conversation. So these worms are very similar to C. elegans. They're also both the parents and the progeny of the parasitic adults. So the parasitic adults survive for many months at host body temperature. The free-living adults can live for maybe 2 days at body temperature. We do not know why one life stage can survive at our body temperature and the others can't. Excitingly, that difference maybe suggests that this is something that turns on once the worm gets inside the host. And that's exciting because an ability that's acquired is maybe an ability that we can target in order to develop novel therapeutic strategies. Another aspect of the question of how do worms get inside the body and live inside the body is, why are these worms ending up so specifically in the small intestine? I should say it's not just the small intestine, it's a very particular part of the small intestine, the duodenum. And we have no idea the answer to that. In fact, for any parasite that is inside another organism, we don't know why these various species prefer the organs and the locations that they do. There's many species that have incredibly specific tropism for a specific location in the body, and we have no understanding for any of them how they're able to decide where to stay.
That is pretty impressive because, I mean, I can't get anywhere without Google Maps. So
pretty big in relation to a parasitic worm.
Yeah, there has to be something that we're missing here when we're studying these parasites, because and it's a fundamental question, and it is fascinating. How do they get, malaria gets to the liver, right?
Absolutely.
About a worm here, he's going to the intestine.
And I know that there are some very talented researchers who are starting to consider this question and to try to understand how might we, in a laboratory environment, tackle these sorts of things experimentally.
So we've talked about some of the assays you can do behavior-wise, and, we're understanding now that it's really important for these worms to get where they need to be in the human body. But how do you study the neural function? Like, are you watching neurons change or respond in some way?
That's right. So we, again, are stealing a lot of, let's say, borrowing a number of techniques from the C. elegans field. And like C. elegans, the Strongyloides worms are optically transparent and they're genetically tractable. Again, this is because of that free-living life stage that enables us to generate transgenic animals. And in the case of neuronal questions, what we can do is express functional proteins in neurons of interest. These include things like calcium indicators, which are these genetically encoded molecules that fluoresce, and their fluorescence gets brighter when neurons are active. So this allows us to directly image the responses of individual neurons or sets of neurons to different sensory cues, such as a thermal ramp that we can deliver to these worms. In addition, in recent years, we've been able to develop techniques for silencing sets of neurons, for targeting selectively different genes inside the genome. That's through a lot of hard work that was done to generate protocols for CRISPR-Cas9 mediated mutagenesis in Strongyloides species. And we're continually working as a field to generate new tools that we can apply in order to ask more and more precise questions.
So have you, for Strongyloides, have you figured out the thermosensor molecules? Are those known? I'm thinking, you mentioned CRISPR-Cas9, so now I'm thinking, well, you could knock them out one at a time and find out, but...
I have a project for you, and I have a project for you, and I have a project for you.
That's great. It's a great project. We've partially done it already. So in Strongyloides, again, like C. elegans, the primary thermosensory neuron is called AFD. And in AFD, there are receptor-type guanylate cyclases that act to transduce thermosensory signals into neuronal activity. The pathway for this is worked out fairly well in C. elegans, and so far we're moderately assuming it's similar in Strongyloides. And the basic setup of it is you have these receptor-type guanylate cyclases that are on the membrane. They act to generate cyclic GMP in response to increases in temperature. That cyclic GMP then activates the cell through cyclic GMP gated channels that are also located in the membrane. And so the activity of those channels lets positive ions into the cell. Positive ions in the cell means the cell is active. So one of the things I did as a postdoc in Alyssa Hallam's lab at UCLA was to take these newly optimized Strongyloides CRISPR-Cas9 tools and apply them to knocking out one of the subunits in those cyclic GMP gated channels. It's this gene called TAX4. And we found that when we knocked out TAX4 in the infective larvae of Strongyloides stercoralis, we could completely prevent them from displaying this heat-seeking behavior.
Okay, very cool.
I feel like I've learned so much today, from evolution to neurobiology to behavior, parasitology.
For me, that's one of the most wonderful things about working in this field, is that it's a chance to bring a real interdisciplinary approach to a major neglected disease and a species that has real importance for global health.
Yeah, I kept thinking about that, and I kept thinking about, like, how do you stay on task sometimes? Because there are so many things that you can look at or questions you can answer. Like I just, I'm bringing in the different models and then comparing, and
we certainly have a lot of work to do.
It's good to be busy.
Our guest today is Dr. Astra Bryant, assistant professor in the Department of Neurobiology and Biophysics at the University of Washington. Astra, thanks so much for being with us.
It was an absolute pleasure.
Links to additional information about Dr. Bryant's research can be found on our Instagram account at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast. And if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, which we are not using today. However, we still thank them. And special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.
Ta-da, ta-da.